Okay, I have the sign, so let's start with today's interview. Hello everybody to a new edition of the bioRN Member to Member session. First session in 2025 and 17th edition so far. And today I'm very happy to introduce HS Analysis to you, a member of the bioRN network whose expertise lies in professional software and hardware systems, as well as the technical infrastructure for automation in pharmaceutical and material science laboratories, biomedical research and diagnostics. And I'm very happy to welcome Sergey Beniaminov, founder and CEO of HS Analysis. Welcome here in our broadcasting studio.
Well, thank you, Stephanie.
Wonderful to have you here. So, Sergey, please introduce yourself and how you became part of your founding story.
Yeah, it was a really interesting time. So it was 2050, 2060 as I founded the company HS Analysis.
Mm-hmm.
And the most important thing is by the founding such a company is you have to bring all your passion and all your ideas in a common goal. And it is really exciting time, and we were specialized on deep learning technology for automated detection and image analysis in that part. And the starting time, it was not like just straight, it was a little bit high and down stuff, and I got great support of my family, for example, which is really important when you're founding a company. Especially my wife supports me a lot and continuing supporting till today, as well as my colleagues and my team, which is really great. And we are researchers, actually, researchers with the aim to create deep learning based assistant for pharmaceutical industry and diagnostics.
Okay, and you studied economics at the KIT in Karlsruhe, right?
Correct.
Okay.
Yeah, so I'm a statistic guy actually then, and understand multivariate statistics stuff and math actually, and deep learning and artificial intelligence is some kind part of this, and it's the next step to the classical statistical methods actually. But I was focused on management of modern medical technologies as well, and entrepreneurship at KIT. We had some courses there, and it was great fun, and actually I got there good marks as well, so that's maybe was the reason.
Great, wonderful. Good marks is
good marks is
a good
always
sign.
good,
Yeah. So let's go and let us talk about the company HS Analysis or HS Analysis. Would you like to tell us shortly about the story of the company? When did you found HS Analysis? What was your motivation and the time? And what is the company mission today?
Yeah, so as I mentioned before, it was 2050, 2060. Official founded was it in 2060, and actually it was driven by the company size, because they discovered our passion at KIT, and then we got the possibility to sell directly by the founding of the company our products actually to Max Planck Institute and one of the biggest pharmaceutical company here in Germany. And that was the start. So we went then to the laboratories there, to Tübingen, to MPI, and then to Wuppertal, and then we created like a software, a customized software for the researchers there in histological labs there. And it was actually the start for one direction, nephropathology, and the other is neuropathology. And till today, we are really experts exactly in these two directions.
Great, wonderful. And when we talked earlier, you told me that you strive for close partnerships with customers. which then lead to innovations and have already established this strong partnership. So this is something that you really want to deepen or expand.
Yeah, of course. So really important to understand is we're driving innovation actually, and we want to do one of the best research worldwide with our customers and partners, and to drive innovation alone, it's not the way at all. Collaboration is everything. What helps to bring the common ideas further. And of course, partnerships is really important in that case. So, actually, we are mostly involved in technical part of projects, and our customers are mostly with the medical background there, and bus parts building together something special then, especially special for the for the reproducible and evidence-based research and medicine. And for example, what we are doing with one partner, which is then from Hamburg and from University Clinic Eppendorf there, the group from nephropathology there. We started 2006 here a common way. Until now, I make a last call yesterday with the professor there. Now we has an idea to write a new grant, DFG grant there, and we started there to develop some system which then imports histological slides and automatically analyze some structures in these histological slides. And I think we used to work on this system something about 2 years. After that, the system was running until now it's running, and in different directions. What do I mean by the direction? Different diseases. That's this aim wouldn't be never reached without partnerships.
Of course, yeah.
So that's most important.
So we're going to hear a bit later about those case studies, but you told me also that you are quite closely involved into papers that are written and mentioned there. So this is really something where we can see that strong partnerships are one of the main drivers of your work, especially. Okay, so we also would like to know, or I would like to know, some more information about your products, because of course you have a toolbox that you use when you work with various customers and institutes. So could you give us a close overview?
Yeah, of course. We have some products which are then hardware systems as well as software systems, but our personal consulting, which is really important by building the designs for different studies. And we are experts in automation and automated processing of data, with a focus on images, but we have a lot of proteomics, for example, multiomics and so on. So that means that we understand the data before they're going to be analyzed in our head, actually. And then we consult the doctors from the medical side. Which kind of data should we use and how deep should we use them? Which technology would be used to reach which status of output from this data? And this step is really important because when you choose the correct direction from the start, you save a lot of time. And this is really important. That means one of the products here is HSA Kit. We called it HSA Kit. This is like a box where we have a lot of different modules, and but the same base as deep learning based assistant for pharmaceutical research and diagnostics as well. And we have different modules for the automated quantification of data. But before this automation works, the data should be digitalized. That's why for the image analysis, for example, we have HSA Scan, which allows you, this is like a box which you integrate into your microscope. Without opening it, it's just a box which is going to be positioned on the microscope and creates you the digital images in that way that is optimal for the deep learning procession in the next step, for example. These are two products. We have as well HSA Cases, which is a software program which has then the combination, or it imports all the data which is going to be digitalized and analyzed and combined with clinical data, for example, and diagnosis information and clinical chemistry data and other types of patient data, or in studies, the data that should be saved for the structured way of saving the data over a long period of experiments, which is really important for our customers to have overview of the experiments independent from the personnel, for example, and we try to bring the objective way into the experiments.
Thank you very much for this overview, and you also brought us a video today that describes the workflow, and I would like to encourage you to give some comments on the video we're going to see right now.
Okay, in HS analysis, so the first step, for example, when we have slides which are gonna be for diagnosis purposes, for example, then the doctor's working analog because it's really fast and easy, but we have there some points like it is always going to be subjective. And when the labs bring in the digitalization, this is a long process and costs as well a lot of money. So we give the possibility to start really fast and cheap with the digitalization before the whole process is going to be integrated. You can use HSA Scan, which is a really powerful but useful tool, and you can snipping some single shots from microscopic images, but as well as digitalize the whole slide image, for example, semi-automated. Semi-automated, it means you check your slide and the software digitalize automatically the tissue, but you can use it as well, for example, for cell culture research and digitalize some cells and analyze live versus dead assays, doing stuff like this, and but as well digitalize the whole slide image, like you see it here. This is for routine diagnostic purposes. We created this with one customer, and what you see here is colorectal carcinoma, for example, and the annotations which can be done automatically by deep learning from HSA Kit.
Right. Wow.
Carcinoma, adenoma, and healthy glands in I think in green.
Okay, quite exciting. Thank you very much for the video. So let's step a bit deeper into real application examples. You told us that you really work very closely with the Uniklinik Eppendorf. So that might be probably the first case study. You can explain a bit what is your work there and how it's going to be developed in the future.
It is a really nice collaboration with fruitful results over the years. We are working there with nephropathology, for example, and nephrology as well. And what we are doing there is we analyzing the tissue from kidney and are able, for example, to predict, to detect firstly the compartments of human tissue, as well from mouse or rats, with the same algorithms. And this system gonna be created actually in collaboration from the UKE, from the MTAs there, in collaboration with us, and they use it in their routine, and they can automatically detect the glomeruli there and categorize these, as well as tubuli and peritubular capillars, for example. And especially is you need to use histological stains, but in some cases you use immunohistological stains, and this is a different process. And what we develop there, we go on histological stain, on histological slide PIS, and are able to detect the single cells within the glomeruli and categorize them. And the professor there, he thinks that podocytes are really important for diagnosis of EGA-associated glomerulonephritis, for example. And today the podocytes are not really, it is not really necessary to check them all, because and the idea is that we actually understand the podocytes really extremely from the PIS stains without any EHC stains. And deep learning is able to do this by morphology and position of the cells within the kidney, and really exactly. We did some studies together, and we showed that the doctors, which are then manually detect all these, are really near to the deep learning algorithms for this aim, for example. Yeah? And this is like an example that we published several papers, and gonna publish one new paper again shortly. This is with UKA in nephropathology, and really interesting. It was the jump to another expertise there, for example, because the doctors, when something works for them, they're talking about this with their colleagues as well. And at UKE we have the director of diagnostic center there in neuropathology as well. And he said, okay, when you're doing this stuff and it works really for your colleagues, MTAs and laboratory, so easy, and at the end you get your numbers, quantification, and based on the numbers and quantification we create some system, recommender system for this or that, and we will do a lot of visualization actually. There is, for example, really important the interfaces to SAP system, which gonna be changed in the next few years because they don't want to do it anymore. But the interfaces to that and communication with the clinical system in general, or this system, it depends on that. And this construct is working, and in the neuropathology we started then import not the kidney slides, but then brain regions, and detect within the brain regions, for example, beta-AMI, HLA signals, and so on. Yeah? This is fruitful collaboration, for example, with Hamburg. Okay.
Right, wonderful. And if you attendees would like to ask your questions, please type them in the chat and I will ask them later on. So now we move to another interesting project, which I've learned comes from collaboration with some big pharma partners, also part of our network.
Actually we have a really long collaboration with one big partner and from pharmaceutical company, and we are completely integrated in their labs for automated quantification of different mouse models, and they are working on actinium and thorium and radiopharmaceutical lab, and what we are working is on colorectal carcinoma and prostate cancer, especially prostate cancer there. And this collaboration is, we have this collaboration since 2016 as well, with full integration in such lab, and this is actually routine work now. But with another partner, which is a big company from our region here, we switched the technology for membrane detection, HER2, HLA, HER2, PSM alpha, markers, HC markers like these, CK5 and so on. We switched it to other laboratory and so for heart disease there, and we detecting on HE slides myocardiocytes, and because technology is the same, but the applications are a little bit different, and it was really important to develop something which is modular and works, and it should be easily transferred to another question, for example, another aim, and we done it for liver, for example, for quantification of cells in liver and classification of these cells, as well as for heart, and it was really challenging, and I mean the quantification of myocardiocytes, it is one of the complex tasks that I got before, and now we're using there, for example, such supporting mechanism, not only on HE. We won't use fluorescent-based slides as well to bring them together and maybe create then virtual slides. Let's see how it's gonna work, but it's something which gonna be to do in near future.
Right, 2 successful cooperations, and I've just know from our preliminary discussion that you are very interested in establishing more fruitful relationships or partnerships with institutes or departments here in the region. So my question is, what are you specifically aiming for?
So what we could show is that the technology and integration of this technology in some groups for research, for example, in university clinics as well, works very well for the different parties there. And what we learned is how it's going to be financed, for example, here in Germany. And there are ways which allowed both sides to bring the innovation forward. And that's why I want to create more collaboration in this region, because my personal support in some different studies is important, because the vision and the mission are going to be driven by single people, and that's what I would like to do in our region, really personally, and that's and really interesting here is that the technology what we have, the deep learning based assistant for diagnosis and biomedical research, is scalable. That means what I showed before in the video, or you showed before, is the histological images. We started with Charité, for example, to transfer this technology to ophthalmology, which completely different part as histology. and it was working really great. And I was last year at DOG conference there, and the PhD students which were using our technology, I mean, you can imagine, it was a PhD student, medical background, without any technological knowledge in that. And the person was able to create its own deep learning network within the clinic, offline. That means a lot because of the data that they are going to be used, completely on its own. And deep learning based assistant supported this person by creating deep learning network, as well as bringing this network in that direction that she's really need for the data that she got. Normally it is a really complex process. You create something, you send to bioinformatic guys, again back and so on, and at the end you don't have the quality that you would have that. And then you take something that you have and then you publish, for example, and so on. And here we got some result from this one person in collaboration with colleagues, of course, but it was fully great of quality that day. And she was presenting it at DOG there, that she was able to train within some couple of weeks her own deep learning network, and she was understanding. And I was allowed to make a short presentation there as well with other. Actually, I was the only one who was not the director of a clinic there. Yeah, it was something special actually for me.
Definitely, yeah. And talking about ophthalmology, you told me also that you have access to the largest ophthalmology registry in Germany of NAMD patients and various other. Is that right?
Yes.
So what opportunities are there for new customers to utilize this data?
Yeah, for example, what we got really huge data samples from MAD there from the patient, and what we were able to detect is fully automatically on fundus cameras, for example, without all the multi-imaging center equipment there, only using by Nikon or Zeiss. These were two cameras which are going to be used there for our experiments. If it's tumor or no tumor, or the grade of the tumor after the radiation, for example, fully automatically and using only optos cameras. That means this step allows you to do diagnosis in the regions where they do not have some professional imaging centers like we have it in Berlin, Kassel, Munich, and so on, for example. And we can bring this innovation to other countries, where the medical infrastructure is not so high professional like we have it here in Germany, for example. That brings my motivation, of course, higher, and helps developing such algorithms, for example.
Thank you. So before I start asking you questions from the audience, one other question is, you're part, of course, of a strong network, bioRN. So how can you help our member network, and what can you offer them? Yeah. So
what we can do is the partners from the network could write me an email if, for example, they can ask bioRN and or talk to me, and when we would support some histological labs or biopharmaceutical industry with detecting some really difficult or standard morphologies there from images or spectral data, for example, as well as creating common grants for the academia and bringing really easy workflows into the pharmaceutical laboratories by using our software, which can be then used by all the MTAs, and you don't need to do their PhD, for example, to using the software. It was creating with kids in the ergonomy And we say there, when the kids can draw the images and draw the cars and trees and train its own deep learning networks, actually, yeah, it was done, then it can be done in the laboratories as well for the new questions, for example, there, but as well using the existing networks, and we have 300 different deep learning networks, yeah, created for the pharmaceutical laboratories, and then they can use it just by one click or even 0 click for the data. And that's what we can do together here.
Great. So, and how can other members support you?
Yeah, they can talk to us, to HS Analysis, and ask where we can support them actually in the automation of analysis of biomedical data.
Okay, wonderful. So let's come to the question, because time is running. Question 1 comes from Daniel Pöckel. Do you have optimized AI detection algorithms or models for identifying cellular or structural patterns in several individual organs to recognize tissue-specific patterns, like kidney, brain were mentioned? Further, what is your coverage of individual diseases?
Yeah, of course we've done it, because it's the most interesting part of my job actually. For example, we got a founded project and from CIM, that being the car, and what we can there, we had non-small lung cancer carcinoma, and we were analyzing the NRF2 KEAP1 interaction and creating a kit, for example, for the drug resistance purposes there. And we had to deal there, of course, with cancer cells and non-cancer cells in this type of issue and that type of Platten carcinoma and so on, as different types of carcinoma. As I mentioned before, brain or colorectal carcinoma, to detect automatically carcinoma, adenoma, and healthy glands and the grade of it, or for example, breast cancer and HER2, Ki67, CK5, HE, and so on, also bring them in combination. So actually, we used all the types of organs that are existing, because we're doing deep learning, and we're doing only this since 2060 full time, here in Karlsruhe, and
So, okay, thank you very much. The second question comes from Claudia Pande. Can you integrate open source pre-trained AI models such as they're offered by, for example, bioimage.io?
Yes, of course. That's what we do. And it is important when you start a collaboration not to develop everything new. It is important to understand the status where we are now and understand the problems or showstopper, because by using such tools like QPass, I like this a lot, for example, QPass, but that's why my company is existing in histology, for example. We started there where QPass ends. And when you want to do one of the best research worldwide, then you don't need to use the software solutions which are limited. And you will reach this understanding then later by using the software after a few months, even years maybe. And what we're doing, we completely integrate the open source solutions into our solution, but we never, what we doesn't want to do is to, we're publishing our knowledge as well in papers, and we're publishing the algorithms which we're using and technology, that what we're doing, because we want to share the knowledge that we created in an academic way, but we don't push the code open source. That's what we doesn't do actually, but we're integrating all the even already trained deep learning networks. Yeah, and sometimes it makes sense to create it new when the actually deep learning network open source has limitations. To optimize this mostly is more difficult than to use ones from our part, and we have their modern technologies developed which are then running completely easy and train the processes new actually. Then you have 3 days train time and you got the result that you want without a lot of validation.
Thank you very much. One last question popped in. It's again Daniel Pöttl, how extensively did you train your models? To cover diseases broadly, you would need probably dozens to hundreds of individual H&E images.
Yeah. Okay. What we are not able to do is train a deep learning network which can do everything from one organ, for example. That does not work actually. What we're doing in our software, we're building such modules, and they are completely separately from each other, but at the end you can bring them together. And that's what we're actually doing. And this is the software. This was the most important part of development, because I developing this software, actually I don't code anymore, but I'm software architect. And that's what I can do good. I'm really worst software coder, but software architect is okay. And what we've done, we created like train station with different trains, and I know which train going which direction, and then we train a deep learning model for one train, and then this deep learning model for this train is able to detect something for glomerulonephritis and pus stain, for example. The next train doing some for ANCA associated glomerulonephritis, for example. The other EGA associated, yeah, and then you run this when you need this actually, and at the end you can bring them together. For example, you can add toxicological factors to this, or hypertrophy factors, and use all the other information which needs to be used in this process actually. Yeah? That means we are not able to train a big deep learning model which can do everything, but we can use single deep learning models, 10. Even in neuropathology, we created 10 to 15 deep learning models, which are then working simultaneously and parallel to each other. That's the way what we're doing. I hope I could answer this question.
Well, thank you very much. We are almost at the end, but if you would like to extend the discussion, of course, you can contact Sergey directly and get into probably a fruitful cooperation. So, thank you very much, Sergey.
You're welcome.
It was great to have you here today.
Thank you for the invitation.
Thank you very much. And for the audience, I would like to mention that this interview will be soon available on the website, of course.